Damn.
Okay, well, I think we can go ahead and get started. I think Nicole will not be here today, so thanks everyone for coming. It's a pleasure to introduce my student, Wyatt Paltzer. He's going to talk about the other side of what we do in the lab. I guess last week Rebecca talked about innervation and how to improve our development and function. Wyatt will be talking about our interest in regeneration and the mechanisms that guide regeneration, so that hopefully we can turn on these mechanisms in the adult heart. So Wyatt, take it away.
Yeah, thank you for the introduction, and thank you all for being here. Today, I will be giving my talk titled Defining the Role of Mechanistic Target of Rapamycin Complex 1, or as I'll refer to it, mTORC1, during heart regeneration. So we're really interested in heart regeneration due to the disease condition heart failure. And so that's shown really well here in these two videos. With the one on the left here, you can see the muscle of the left ventricle here is really well-defined and really strong in comparison to the heart with heart failure here on the right. And so what you can see in the normal heart is that when it beats and pumps blood, you can see that it's functioning very efficiently and very strongly able to eject the necessary blood to the rest of the body to properly function and provide the body with oxygen. That's in stark contrast here to the heart with heart failure on the right, where you can see with that thinning of the left ventricle, it's significantly weakened and no longer able to function as efficiently as a healthy heart. So this leads to a number of complications in patients with heart failure, and it's quite a debilitating disease. Furthermore, it's also a very common disease, with roughly 6 million patients currently diagnosed with heart failure, with that number only expected to rise in the coming years, with an estimated 8 million patients concurrently diagnosed with heart failure by 2030. The severity of this is also a big concern because there's no true curative therapeutic for heart failure. Once the heart tissue has been damaged, the only treatments can help mitigate some of the symptoms, or if there is something to help the heart pump blood, it only lasts for a certain time and requires very serious invasive surgeries. So with that all in mind, the field is really turned towards heart regeneration as a possible way to identify curative therapeutics and come up with better treatments than what we currently have. So heart regeneration for a while has been known to occur in some species such as zebrafish and some amphibians, where whether a heart injury occurs at early neonatal stages all the way to adolescent or adult stages, those hearts can completely regenerate with little to no sign of any heart injury ever occurring. That's in stark contrast to much more complex species such as humans, which completely lack any natural regenerative capabilities of the heart at adult stages. However, there are some species such as mice and other mammals that have the potential to regenerate lost heart tissue at early postnatal stages. However, as they mature and develop, that ability to naturally regenerate the tissue will fade. What this does is it helps us be able to study processes that control heart regeneration when it's naturally occurring and identify whether or not these processes could be stimulated to reactivate heart regeneration during time periods where that regeneration does not naturally occur. So in order to do this, we'll use models of heart failure, which specifically our lab uses the myocardial infarction model. And the way we'll do this is comparing heart regeneration in the neonatal mouse and the adult mouse to examine what processes can regulate that heart regeneration. So you can see here, both the neonatal and the adult mouse model begin at surgery with a ligation of the left coronary artery, which you can see here and here, ligating at roughly the same position in both the neonate and the adult heart. 7 days post-surgery, the neonate and adult will begin to have some scarring below that suture site due to the blood flow to that lower left ventricle being blocked by the suture, leading to a myocardial infarction. However, where the two models really differ is 3 weeks post-surgery and later, where in the neonatal model, you can see that that scar tissue that began forming at 7 days post-surgery has completely resolved itself, leading to a healthy myocardium, where you can see little to no sign of any injury occurring except for the suture site. That's really showcased here in this trichrome stain, which lets us analyze the heart for healthy tissue. And what you can see here in the lower left ventricle here is healthy myocardium showcased by that red staining, really showing that not only is the tissue undamaged, but there's little sign of any injury ever occurring. That's in stark contrast to the adult heart, where that scar that began forming at 7 days post-surgery has not resolved itself and instead led to a large scar zone formation, which then when you look at the trichrome staining, analyzing the health of that myocardial tissue, you see that instead of having a well-defined myocardium here, they instead have this fibrotic blue stained tissue, which is, again, just fibrotic tissue that has replaced that damaged myocardium and led to a significant weakening of the heart overall. So using the neonatal and adult myocardial infarction models, we can begin to understand these biological processes regulating heart regeneration. So as initial studies done by the field looked into these processes, they really identified that cardiomyocyte proliferation is the key biological process regulating heart regeneration. And so essentially, neonatal cardiomyocytes are highly proliferative and have this high proliferative capacity, but over time that diminishes as cardiomyocytes are exposed to different damaging sources along with just general maturation. One thing that comes along with that general maturation is a metabolic preference in substrate. So early neonatal cardiomyocytes really prefer glucose as a metabolic substrate preference, relying highly upon glycolysis as a source of energy. As the heart matures, it goes through a metabolic switch, switching from glucose as a primary energy substrate to fatty acid oxidation. What this does is it changes the way that the heart will utilize energy to make it more efficient. And as this switch occurs between postnatal day 1 and day 7, which is roughly the same time period that the neonatal heart can no longer regenerate. So as this neonatal switch in metabolic substrate preference occurs, as I mentioned, at the same time as that switch for heart regeneration, we really wanted to observe whether or not if this could be reversed and switch the adult heart back to relying upon glycolysis in order to stimulate heart regeneration. So previous work done in our lab by a former postdoc showed that if you could switch the heart back to utilizing glycolysis as a primary energy source instead of fatty acid oxidation, that you could in fact promote heart regeneration. So this was done by using the succinate dehydrogenase competitive inhibitor malonate, where when animals were treated with malonate, we saw a deprioritization of the citric acid cycle and fatty acid oxidation, while we saw an increase in the use of glycolysis and glucose metabolism, showing that competitively inhibiting succinate dehydrogenase, we could successfully reverse that metabolic switch in the adult back towards glycolysis. The outcome then on heart regeneration is showcased here on the right, where the adult hearts that still relied upon fatty acid oxidation and treated with our vehicle control saline had a large scar formation in the left ventricle that is shown all the way from just below the suture site all the way down to the apex, really showing that those hearts would be a signature of heart failure. However, in the succinate dehydrogenase competitively inhibited hearts, in animals treated with malonate, what you can see is there's a little scar right by the suture. However, in the rest of the left ventricle, there is little to no sign of any damaged tissue, showing that when we could promote glycolysis, we could in fact promote heart regeneration. So this work done by that former postdoc in lab really gives credence to when we can reverse that metabolic switch, we can in fact promote heart regeneration. So with that in mind, we wanted to begin to identify some signaling pathways that we could use to promote glycolysis to stimulate heart regeneration and ultimately possibly identify novel therapeutics. So with that, we then did some dive into the literature to see if there was any good candidate pathways, and we came across some work done by some collaborators now at Johns Hopkins University that showed that mTORC1 can regulate metabolism after cardiac injury. And so what those collaborators did is they used single amino acid substitutions of the mTORC1 genetic regulator, TSC2, to either inhibit mTORC1 or hyperactivate mTORC1. And when they used the genetic model to inhibit mTORC1, they saw an even further decrease in glucose metabolism by adult hearts after injury, and an even further increase in fatty acid metabolism, kind of driving the normal adult metabolic utilization to extremes. However, when they hyperactivated mTORC1, they saw the reverse. And just like we did in our previous work, they essentially reversed that metabolic switch, where they then promoted glucose metabolism and downregulated fatty acid metabolism, really showing that mTORC1 is positioned as a key regulator of cardiac metabolism that we could possibly modulate in order to promote heart regeneration. So the first thing that we wanted to examine, though, after honing in on mTORC1 as a key pathway for this, is whether or not mTORC1 is even required for neonatal heart regeneration. So the first thing that we did is we wanted to examine whether or not mTORC1 functions differently during heart development versus regeneration. And so we examined two key mTORC1 substrates, the S6Ks here, examining P70S6K specifically, and 4E-BP1. And we saw stark differences between developmental and regeneration. So what we saw essentially is in both P70S6K and 4E-BP1, we saw high levels of phosphorylation at early developmental time points, specifically P2 and P8, and then a decrease at P29, and then adult stage is not shown, showing that mTORC1 is really acting upon these substrates early in development. However, we saw almost the opposite in the regeneration time series, where there was little to no phosphorylation of the P70S6K and little phosphorylation of 4E-BP1 one day post-surgery, which does correspond to the P2 developmental time point. And then instead of decreasing over time like we saw in the developmental time series, these substrates actually increase in phosphorylation over time, showing that mTORC1 is functioning differently under regeneration conditions compared to developmental conditions, showing that mTORC1 may be a key regulator of heart regeneration. So we showed that mTORC1 functions differently during development and regeneration, but we really wanted to examine whether or not mTORC1 was actually required for neonatal heart regeneration to occur, or whether or not it may not be key and regeneration can still occur even if mTORC1 is inhibited. So to do this, we used this experimental timeline where neonatal mice would undergo a myocardial infarction surgery at one day postnatal. And then on days 4, 5, and 6 post-surgery, they would be treated with either a vehicle saline control or with our mTORC1 inhibitors, rapamycin and everolimus, which are very similar inhibitors, but we wanted to use two different ones to make sure we weren't getting inhibitor specificity. And then we collected for analysis at either 7 days post-surgery or 28 days post-surgery, depending on the analysis that we would be conducting. So when asking whether or not mTORC1 was required for neonatal heart regeneration, we were able to show that mTORC1 inhibition does prevent neonatal heart regeneration. And you can see that here in the saline-treated hearts on the left, where there is little to no, unfortunately, the projector doesn't really do this justice, but little to no fibrotic tissue shown in blue, with mostly healthy myocardial tissue shown in red. Whereas in the mTORC1 inhibited hearts, they both have this large scar formation and reduction of the healthy myocardial tissue, showing that the remaining cardiomyocytes that were undamaged by the myocardial infarction did not repopulate that damaged tissue area, leading to a dense scar area, and also they're slightly enlarged from cardiac hypertrophy. This cardiac hypertrophy is then supported through a heart weight to body weight ratio, where there's a significant increase in the heart weight to body weight ratio of the mTORC1 inhibitor everolimus treated hearts, with the general trend in the rapamycin treated hearts, albeit not significant. So now that we know that mTORC1 inhibition does in fact prevent heart regeneration, we were wondering if it was due to preventing proliferation, as cardiomyocyte proliferation is key to heart regeneration occurring. So in order to assay cardiomyocyte proliferation, we use the common mitosis marker, phosphohistone H3. And what we saw is, again, a significant reduction in the amount of proliferating or mitosing cardiomyocytes in mTORC1 inhibited hearts. And so you can see on the left, the way that we do these assays is we'll stain a whole heart section, and then we will quantify every single positive cardiomyocyte across that whole section. So this is just a representative cardiomyocyte of what we would be quantifying here. But so when we quantified across multiple sections and multiple biological replicates, we see a significant decrease, as I mentioned, in the total number of mitosing cardiomyocytes, showing that when mTORC1 is inhibited during heart regeneration, it is decreasing the proliferation of these cardiomyocytes. However, as cardiomyocytes are a multinucleated cell population, we also wanted to examine whether or not they were actually just, this mTORC1 inhibition was preventing the cardiomyocytes from becoming multinucleated, or if the mTORC1 inhibition was actually preventing the cardiomyocytes from dividing and generating new cardiomyocytes to replenish that damaged cardiac tissue. So in order to examine that, we used the cytokinesis marker Aurora B kinase, an assay very similar to that pH3 staining. We again stained the whole section and then quantified the number of Aurora B positive cardiomyocytes across that section with a representative Aurora B kinase positive cardiomyocyte here on the left. And again, what we see is a significant decrease in the amount of Aurora B kinase positive cardiomyocytes in the mTORC1 inhibited hearts, showcasing that not only is mTORC1 inhibiting the heart regeneration, but it's doing it through preventing the cardiomyocyte proliferation, essentially blocking cardiomyocytes from generating new cardiomyocytes to replenish that damaged myocardium. So the last thing that we wanted to check as kind of a confirmation test, making sure that we are in fact seeing a decrease in the generation of new cardiomyocytes, is the cross-sectional size of the cardiomyocytes, as if the cardiomyocytes that are remaining should be enlarged in comparison to the controls because they're generating new cardiomyocytes to replace the damaged myocardium, bringing down the average cardiomyocytes cross-sectional size. So on the left, we just have a cross-sectional cardiomyocytes that will be used as, for example, to quantify here, using a cell membrane stain, where in this instance we use wheat germ agglutinin. And then we'll just measure the size of individual cardiomyocytes. And across measuring hundreds of cardiomyocytes across multiple sections and multiple biological replicates, we can see that there is in fact a significant reduction in the size of the saline-treated hearts, showing that those ones did generate new new cardiomyocytes to replenish the myocardial tissue, where the rapamycin and everolimus mTORC1 inhibitor treated animals did not generate those new cardiomyocytes, and thus had individual cardiomyocyte hypertrophy to go along with the cardiac hypertrophy. So the main question, though, that we want to ask when studying regeneration is whether or not we can ultimately stimulate regeneration in instances that regeneration does not normally occur. So we're really interested in whether or not the hyperactivation of mTORC1 could promote heart regeneration in situations where that heart regeneration would not normally occur. So this is really where my future research is going to go. And I'm going to examine whether or not the mTORC1 hyperactivation could promote heart regeneration. And I'm going to use those genetic models that I talked about earlier. Specifically, I'm going to use the TSC2-SC to examine whether or not the inhibition of mTORC1 will block the genetic inhibition of mTORC1 will block heart regeneration, similar to what we see with the pharmacological inhibitors. And we believe that'll function because it's going to decrease the reliance on glycolysis that is normally there and increase the fatty acid oxidation reliance in the neonatal heart, which is essentially reversing the switch in the neonates. So the neonates will prioritize fatty acid metabolism compared to glucose metabolism. The one that we're really interested in is this TSC2-SA allele, which we believe can hyperactivate mTORC1, that our collaborators showed after cardiac injury that it would increase the reliance on glucose metabolism, therefore reversing that metabolic switch and leading to hopefully heart regeneration. So with that, it's a good time to talk about my rigor and reproducibility, which, as always, with any lab-based work, keeping good procedural and experimental notes is key, along with genotyping all mice and using positive and negative controls. But something that's much more necessary for my work that we haven't really talked about yet in the seminar series this summer is maintaining good surgical accuracy and reproducibility, as my models heavily rely upon my ability to do that efficiently and accurately. So some things that I'll always do is I'll always make sure that I can visualize the LAD before I ligate, making sure that I'm actually seeing the vessel that I want to ligate, and then I'm ligating in the same location just above this branch point here. And then also I'll watch that artery and make sure that after I ligate it, it actually blanches, and I can see that I have prevented blood flow to that lower left ventricle, really showing that I'm actually inducing a clinically significant MI. Furthermore, I'll also carry this out to the actual analysis stages, where I will perform trichrome stainings or other ways of analyzing whether or not I did induce a myocardial infarction properly, and examining that I'm getting regeneration when regeneration should occur according to the field, and not getting regeneration when regeneration should not occur. Essentially making sure that my surgeries are meeting field standards. So then just a short summary of my talk. I showed that mTORC1 functions differently during development and heart regeneration, positioning it as a possible regulator of heart regeneration. I then showed that inhibiting mTORC1 can prevent heart regeneration, resulting in cardiac hypertrophy and scar formation due to inhibiting that cardiomyocyte proliferation. And then also, in the future, I would like to investigate mTORC1 hyperactivation as a mechanism to stimulate adult heart regeneration. So with that, I would just like to acknowledge a couple of people: Ahmed for being a great mentor, faculty mentor, and then a former postdoc, Ji Young, and a current graduate student, Rebecca Salmon, for really helping me get going in lab and getting to where I am as a graduate student. I wouldn't be there without them. So with that, I would love to take any questions. Thank you. Yeah.
I have two questions. One is if you have any thoughts about why TOR activity goes down in adulthood. And the second is, so is the model that the metabolism is between TOR and cell division? And if so, how does that work?
So just breaking those two questions down, I'm going to start with the second one, the model between TOR and cell division. So essentially, we think that the metabolism is necessary to help essentially provide enough energy to give the cardiomyocytes the ability to divide. And when they're using the fatty acid oxidation, it's so specific and so specialized that it doesn't quite, they don't have the energy to ultimately divide and break down. So it's kind of, mTOR will kind of segue away from that and drive them back towards glycolysis, which should then promote them dividing as cardiomyocytes. So that's kind of where we think it is like kind of that way. And could you repeat your first question for me?
Oh, do you have any ideas for why TOR activity decreases in adulthood?
I don't have any good ones specifically in the heart. I do believe that we are looking a little bit more at the mTORC1's like stress response specific, I think is really where mTORC1 is going to be key in heart regeneration. I didn't get too much into it just due to time constraints, but the TSC2 amino acid that's mutated is a stress response exclusive amino acid, so it doesn't actually regulate mTORC1 under homeostatic conditions. But yeah, I don't have any good ideas of why that would go down. It could also be kind of, it's still there, but it's at a lower level in comparison due to how specialized the cardiomyocytes get. But yeah, I don't really have anything, sorry. Yeah.
I just have a general question, because in animal models, treating them with rapamycin increases longevity. And I assume that would include improving heart function in someone's age and all of that.
Yeah, so I think that gets more at the stress response too. So because it is well known that treating with rapamycin does increase longevity, but it's also shown that treating with rapamycin will actually worsen heart failure. There was a study done back in about 2010 that rats that underwent myocardial infarctions that were treated with rapamycin after the infarction actually had significantly larger scar sizes. So essentially showing that mTORC1 is for some reason worsening the reaction to it, but that's not super clear. So essentially mTORC1, I believe here, is functioning in more the stress response, and it's able to kick that stress response into gear stronger and really utilize that, versus where it's more, mTORC1 is known to have functions under homeostatic effects, and I believe that's more where the longevity comes in with mTORC1. It's kind of like the two different functions there. Yeah, Chris?
I had a couple of things. Kind of in response to that quick, another way to think about it might be like, on this might be, if it's doing a metabolic switch in state, like glycolysis might have a higher long-term oxidative load, and so you're damaging tissue over time long-term, but it's good for cell division, and therefore for like acute injury, it's better. So like that might be one way to like think about it. If that is the difference here, like it's a maintenance versus injury recovery separation. So nice.
So just 1 second, because when you have a cardiac failure, the problem is the oxygen come back, and a lot of reactive oxygen is being created, and that causes a lot of damage. So you need to stop using mitochondria right away, or you start doing more damage than regenerate or whatever you're doing. That's why most of the cases you start using glycolysis to regenerate whatever organ you're talking about, even the heart. I understand what you're saying, but I think in this case, maybe because it cannot use mitochondria and it still need a lot of energy to duplicate cells, and that's why it's using glycolysis and not, for example, amino acids or other stuff. So the same thing with the malonate that will impact the TCA cycle. You need to cycle the cycle to be able to continue using the amino acids, and that's why you cannot shift to protein. You need to shift to glucose utilization.
Well, my understanding is that it's not just that it's doing glycolysis after injury, it's doing glycolysis during development too.
I know. But when you need to regenerate, which is the case here, you cannot just use glycolysis because you want, but because you need it. You're only forward that will not have anything being damaged with that.
Yeah, but I mean, the condition here is that the adults are never choosing glycolysis. If they're just getting damaged to die.
Yeah. They're still getting damaged, but that's why probably they changed to fatty acid or whatever has a high load of energy to carbon or protein or fat acid or whatever. When they go older. Sorry.
So I had a separate question, which was when you were talking about the mechanisms of mTORC, like you're proposing that it's reducing mitosis somehow directly. Do you have alternative control inputs that you can use to see if there are other things that you can target a pathway? Like, are there alternative ways to target mitosis in these cells, or alternative ways to target the like glycolysis, small like fatty acid metabolism networks that you could use as controls?
Yes is kind of the short answer. No is kind of the long answer. Due to the complication of it and also working with mice, we want to limit kind of the experimental play like that. But we did show that malonate can target it directly, but we also want to look at something that's a little less direct than actually targeting specifically the metabolic pathways directly, but looking at a more key regulator, which is why we moved to mTORC1. But yeah, there's also been look at like CHEK1 that just completely controls cell cycling, and when you inhibit that, you do inhibit heart regeneration. When you do force drive cell cycling in the adult, you can't get heart regeneration.
Which is kind of the whole expectation.
Yeah. So yeah.
Final question, which is a little bit maybe tangential, is why do you think some organisms lose adult heart regeneration?
Yes, I also think it's like partly an organ complexity too. Just off the top of my head, like looking at some of the ones that can regenerate more, like zebrafish have a 2-chamber heart compared to a 4. Off the top of my head, I don't know of any 4-chamber hearts that regenerate throughout life, but I could just not know of one yet. But so I think it's partly a complexity there, along with also just the specialization of the cardiomyocytes. They tend to be larger and other sort of things like that in the mice compared to zebrafish or some of the other species. Along with, I also believe nucleation number is different and increased in like higher mammalian species. Yes, question in the back.
Can you comment about the discrepancy between the phospholipid staining where it looked like it was like 42% of the cells were in mitosis versus the aurora where it was like 8? And do you know the mitotic index around the unical part?
So
What's closer? Because you said there was like cell fusions that were happening with your phospholipid.
yeah, so I don't, and I can't really comment on that. I do know that as the hearts mature, they do become multinucleated up to 4 and 8, and I even think sometimes 16N, or like 16 nuclei. So, but I can't speak to the difference of that. I know it does meet closely general field standards that we've seen in other published work, but I don't know specifically that.
Would you say 8% is more close? I mean, early versus like that. I guess there's a timeline of when it's taken. That's in stitching. But do you count all at the same time the cells in mitosis to create a mitotic index for certain stages of heart development?
Oh, I don't know if that's been done. Ahmed, has that been done?
So at least in the adult heart, the level of turnover is about 1%. It's just too low to replenish any of the massive loss of the myocardium. So it's been done with isotope tracing and carbon dating. So we know the levels of adult development. It's much higher, but we don't have exact percentages, but it's definitely much higher than the 1%, which even decreases as we age to about 0.4%.
I hand it over to you now.
So while we're getting set up, it's my pleasure. My name is Tim Donahue, and I'm a faculty member of the bacteriology department. And it's my pleasure to introduce the next speaker, and that's Ben Hall. As the slide says, Ben is a 4th year student in our lab and came to us from the University of Miami, where he had a lot of experience in research as a triple major in marine biology, biochemistry, and molecular biology. And he expressed an interest in working on metabolic activities that could help society come up with new greener technologies. And that's one of his projects that he's going to talk to you about today. So I'll turn it over to Ben.
Thank you, Tim, for that introduction. Thank you all for being here, and those on Zoom for listening to Wyatt's talk and my talk today. Means a lot to have you with us. So today I'm going to tell you about my project characterizing a broad specificity bacterial lactonase that has come out of some of the work that I've done. So this picture, some of the newer graduate students might not know this, but when I first started here in 2018, in August, there was record floods in Madison. So this is actually somewhere down Mineral Point Road. Terrible flooding. It was like a 100-year flood being described as that. And the reason I want to bring this up right now is that extreme weather events like this are predicted to happen more often in the future because of climate change. So if we look on the left here, we have a map of days every 100 years with greater than 4 inches of precipitation. So, you know, how many days are having heavy, heavy rainfall? 1980s to 2010, spread about the state, but what you can focus on is 2040 to 2060. These are going to become a lot more common due to climate change. And really one of the main drivers of climate change is the increasing amounts of carbon dioxide in the atmosphere. So this is a graph of atmospheric carbon dioxide levels measured by NOAA. And we can see that not only is it increasing over time, it's increasing more quickly over time, which is definitely something that we're worried about when thinking about climate change and is something that we want to mitigate in whatever ways we can. Interestingly, a majority of human emissions comes from the burning of fossil fuels. So burning gasoline to run your cars or transport those Amazon packages you ordered last week, or planes, all of that. So the majority of human emissions actually come from burning fossil fuels. So there's a really great need to reduce our carbon footprint when it comes to fossil fuels in particular. I work with Tim Donahue as part of the Great Lakes Bioenergy Research Center, which is a group of labs here at UW-Madison, Michigan State, and some other universities, really focused on the big idea: how can we turn plant biomass into fuels so that we can use a renewable source of fuels and not keep digging up fossil fuels and releasing more carbon into the atmosphere over time. The way that our lab specifically focuses on this problem is using microbes. So we can use microbes to help us turn plant biomass into fuels and other things. So microbes can produce a variety of useful chemicals. Some of you might be familiar with E85 ethanol. Maybe you've seen it at the gas pump. This is ethanol that's been produced by microbes from corn. They can also produce other more high-value fuels like isobutanol shown below, and a lot of other specialty chemicals depending on what you're looking to create. Now, it would be great if we could just cut up grass and throw it in a beaker and then say go and be done with it, but it's not that simple. It doesn't work like that. We need to add some extra steps in. So really what it comes down to is we have to depolymerize biomass before we can let microbes ferment it. And what that means is our plants, our starting materials are made up of these polymeric compounds, so long chains of sugars and other molecules. And we need to break apart those chains before the microbes can utilize them. So I specifically have been working on a project that looks at this chemical called GVL, or gamma-valar lactone. I'll just be calling it GVL for the rest of this talk. But essentially, this is a chemical that's used in biomass deconstruction. We use GVL to help us break apart those long polymeric chains into small subunits that then we can feed to microbes. So GVL is great at doing that, but when we use GVL in this process, we end up with some residual GVL in our mix of sugars. So even we can recycle a lot of it out, but even after that recycling, we can be left with as much as 2.5% GVL by volume in that mix of sugars. And this turns out to be a problem for us. On one hand, that level of GVL is toxic to many biofuel producing microbes. So what we're seeing here is a growth curve, the final growth density of yeast grown with various amounts of GVL added to the media. So you can see at 0% on the left, it's growing fine, 1% doesn't really care. But once we get into that 2 to 3% range, the yeast are not reaching high densities. They're having trouble growing. And this is true for other biofuels microbes as well. Another problem with that extra GVL is it's a significant unused energy source. So if we think of the chemical energy in that sugary mix that has some GVL in it, the GVL can be as much of a quarter of that available chemical energy, but things like yeast and other microbes, they can't touch GVL. They don't know what to do with it. So that's essentially going to waste, whereas we might be able to utilize that and get greater yields. So there's a need to degrade that leftover GVL. We know it ends up there. We know it causes problems. So we need to do something about it. So that's where my project really started off. A few years ago, a previous member of our lab, Zach Oshlag, found that this alpha proteobacterium called Rhodopseudomonas palustris can metabolize GVL as a sole carbon source. So if we put GVL as the only food in its media, it'll grow a little slower than it normally does, but it still grows. We can make plates that have GVL as a sole carbon source, and we get colonies. It likes to eat this, but my question, or one question that's driving my research, is how is this microbe metabolizing this compound? I'm really just going to talk a lot about what we think is step one for degrading this compound, and that it involves the hydrolysis of GVL to form this other chemical called 4-hydroxyvaline. So when I first started on this project and started thinking about this problem, how is GVL being metabolized by this microbe, I did some searching in the literature, and this reaction is known to happen spontaneously, but on the time scale of months. So if you leave a tube of GVL in water on your bench, it's reported to slowly convert to 4-hydroxyvaline. However, in our bacterial cultures, we know our bacteria is growing on the matter of days. So it's not waiting for this to happen naturally, most likely. Instead, it's probably using an enzyme for this reaction. So I wanted to directly test, does my bacteria have lactonase activity for GBL? So what I did is I measured levels of GBL and 4-hydroxyvalerate using HPLC mass spectrometry. And what I did is I took whole cell extracts from my bacterium and added pure GBL to those extracts. What I saw is over time, GBL was stoichiometrically converted to its hydrolysis product 4-HV when I added extract. In the absence of extract, this reaction didn't really occur. So that tells us, yes, there probably is something in that extract catalyzing this reaction. I did some more digging through the literature to find candidate enzymes that I might want to test to try to identify a lactonase. I found, for example, there was this one enzyme from Bacillus that was shown to hydrolyze another lactone, which only differs from GBL by one methyl group. So I figured that's a great place to start. Let's look at the 2 homologous genes in my favorite bacterium. Testing 2 genes is a really small number, so let's just take everything that has the same protein domain in my favorite bacterium, test those all. And I also found that there was an enzyme in humans reported to hydrolyze GBL specifically. So I looked for a homolog in my favorite bacterium, didn't find anything, but then I used a PSI-BLAST, which lets you find more distantly related proteins, and did come up with one hit. So I ended up with this list of 7. I expressed them all in E. coli and tested the cell extracts of E. coli expressing each of these for activity. In those E. coli extracts, only the E. coli expressing this last protein or this last gene, RPA3624, showed evidence of activity. So that was great. I had identified an enzyme that could hydrolyze GBL. Once we had this, though, I started thinking, why would this enzyme even evolve to have this activity? GBL is an industrial solvent. It's present maybe at low concentrations in some natural environments, but definitely not something you find all over the place. So I thought, let me test a bunch of different lactones with this enzyme to see what we can learn about this specific protein. And I found that RPA3624 is a really broad specificity enzyme. So what you're looking at here is a graph of catalytic efficiency on various substrates. So the y-axis here is catalytic efficiency on a log scale. So we're seeing orders of magnitude difference in efficiency for different substrates. And along the x-axis, you can see the different structures of the different lactones that this enzyme has activity on, including some esters at the very end there. So we can see this enzyme acts on lactones with different ring sizes. We've got 5-membered rings in blue, 6-membered rings in red, 7-membered rings in green. And we also see a sort of trend here for the lactones that have these long hydrophobic R groups, like those long hydrocarbon tails or an aromatic ring, seem to have higher catalytic efficiency values compared to the other substrates. So I want to take this moment to talk about my reproducibility and research slide. And thinking about this, I did a lot of enzyme kinetic experiments. So I got a lot of data that looks something like this, testing various concentrations, measuring activity. And sometimes you end up with error bars that are bigger than your little scientist mind wishes you had. And so I'm sure many of you or all of you have been taught many different strategies to reduce variability in your replicates, like use the same protein prep if you're doing protein work, or use the exact same buffers when you're growing something, testing something, do all your replicates on the same day. I'm sure you can name many others that you do on a daily basis. I want to challenge you all to consider ignoring that for a particular reason. So why would I bring up such a heinous idea in genetics colloquium? And I think it comes down to, for me, thinking about what does reproducibility mean? So think about the last experiment that you all did. Maybe it was today, maybe it was yesterday, last week, a year ago. What would it mean for that experiment to be reproducible, for the data you got to be reproducible? What I think this means is that someone else in a different lab at a different time can follow your protocol and get results that are not significantly different. So to me, thinking about reproducibility means thinking about how can I capture the variability that someone else should expect if they are reproducing my experiment. So someone else who's going to be doing this in a different time, who might be repurifying an enzyme, remaking these different buffers that they're using in this, but following my protocol. So for my own work, this looks like, using protein from separate purifications as from separate cultures as my biological replicates, remaking reagents as appropriate, and still using that data in my analysis. And an important point I also want to bring up is accepting realistic variability, even if it doesn't, look good to what you wish you had in your little scientist heart. I want to bring up that one thing that's important to think of is repetition in pursuit of lowering your error bars or reducing your error bars is kind of a form of data cherry picking. So I will challenge you all to, if you're thinking, oh, let me just do this experiment one more time to try and get those data bars down, stop for a second and think about, is this the appropriate thing to do, or does this, fall into the realm of data cherry picking? So something to think about, and something that I thought about a lot when doing this work with all these enzyme studies. So thinking back to my particular enzyme, one thing that intrigued me was it's annotated as a gluconolactonase. That's what NCBI says, which would suggest it has activity on gluconolactone. But I could not detect this activity. I tested it multiple times in different types of assays, but I never saw it. So after digging into this more, I found that it seems like the gluconolactonase annotation is actually describing two different types of enzymes. Enzymes that act on gluconolactone and other sugar lactone molecules, or enzymes that act on these more hydrophobic looking lactone molecules. So what I did is I looked at the amino acid sequences for various described lactanases, and we created a phylogenetic tree, and it turns out that this tree groups these lactanases by their substrate specificity that has been reported in some way. So we can see that in the blue groups on this tree, they're all more closely related to each other than they are to any of these sugar lactanases, suggesting there is something about these sequences or these enzymes that have evolved to act on more hydrophobic lactones. Notably, this grouping is not driven by just what domain these enzymes are from, because we can see even within the blue group, the hydrophobic lactanases, we have enzymes from eukarya, gram-negative and gram-positive bacteria. So what else can we learn about this enzyme? We looked at substrate specificity, but we haven't really thought about how does this work? And to do that, we really wanted to turn to using structural data. So I worked with Craig Bingman in the Brian Fox lab here on campus. Craig was a huge help and did all of the crystallography and X-ray work. He got these really cool pictures, and I got to actually see my protein crystals under the microscope, which was a lot of fun. Highly recommend if you're ever doing crystallography. And so Craig got these crystals, blasted them with X-rays, and was able to get us a structure. And what we see from this structure is that this enzyme is a 6-bladed beta propeller. It has a couple things bound to it. It has a calcium shown as the green sphere, kind of right in the center. It has a phosphate molecule stuck to that calcium, and it has a sodium ion further down in the center of this molecule. We knew that, or we hypothesized there might be a calcium there just based on sequence-related lactanases, but we wanted to make sure that it actually was a calcium. Metal-dependent enzymes are notorious for having multiple different metals or preferring one metal, but sometimes if you crystallize your protein and calcium-containing buffer, then you might get calcium even if the enzyme doesn't like it. So we wanted to make sure that the calcium was actually there. So I ran ICP-MS on this, which just measures metal concentrations. And we do see consistently, we have basically exactly one molecule of calcium for every molecule of protein we have. And notably, we don't detect other common metals that are in enzymes like magnesium or zinc or iron. So we're confident that that is a calcium, and this is a calcium-dependent enzyme. Also, we know that calcium is important for activity because when we add calcium chelators, we get severe reductions in activity. So something is definitely going on near that calcium that is involved in catalysis. So how does catalysis work? We know the calcium is important, but what else can we learn? In thinking about lactonase enzymes or enzymes catalyzing hydrolysis of lactones, the field mainly looks to identifying an amino acid residue that steals a proton from a water molecule, which allows that water to attack your lactone. So my goal was essentially thinking about how can I identify what amino acid residue is responsible for this activity. And the first thing I did was assay enzyme activity at different pH values. So we see catalytic efficiency on the y-axis on a log scale measured at different pH values for this enzyme. And we can see there's a steep drop in activity that occurs around pH 5.5. So the placement of this acidic shoulder in this curve suggests to us that an acidic amino acid like this aspartate or glutamate is probably involved in catalysis rather than a histidine. If a histidine was involved in catalysis, other groups have shown that this drop in activity occurs more towards the pH 7 to 8 range. So we do think it's an acid involved. So we know that an acid is involved, or we think an acid is involved. We also know the calcium is important from our previous assays. So let's look at the calcium and look for nearby acids. And sure enough, we do have an aspartate D229 next to the calcium and a glutamate E15 next to the calcium as well. So the next thing I did was make amino acid substitution mutants of this enzyme and assay those variants for activity. And when I change this E15 to an aspartate, so I'm changing it, I'm changing the shape of that acid. I lose activity, but I still can detect some working enzyme. And we know that this enzyme is also still binding some calcium because I did ICP-MS on these variants as well. However, when I make the reciprocal change in D229, so when I substitute D229 for glutamate, again, it's still an acid, but now we're changing the shape of that acid. I can't detect any activity. So we're severely messing up the enzyme somehow, even though we are still binding some calcium. So we were always worried about maybe we're just completely losing calcium, but we do still see some calcium in these mutant enzymes. So this data to me is suggesting that the positioning of that D229 residue is more important for activity than the positioning of that other acid E15 is. So after we did this, Craig and the Fox Lab actually had a pretty brilliant idea, which was to soak our crystals in GVL. So when you do X-ray crystallography, you need to put your crystals in a cryoprotecting solvent so that you can get them really cold to be ready for getting blasted with X-rays. And it turns out GVL is a cryoprotecting solvent, but it's also a substrate for us. So that means we can add tons of GVL to our enzyme and basically force GVL to enter what we hope is the active site and then get that data in X-ray crystallography. What we do see is in our crystals soaked with GVL, we do see electron density in the active site right next to that calcium, which is kind of shown by these meshes in these figures. This density doesn't match great with GVL by itself or the product by itself, or even a predicted tetrahedral reaction intermediate by itself. So what we think is happening here is the enzyme is actually undergoing catalysis to some degree in the crystals. So we have a mixture of all three of these different things in our crystal, which is kind of leading to this more amorphous electron density that we see in the active site. But we can still model each of these pieces into that density to get an idea of, if it was just GVL there, where would it sit? What contacts would it be making with nearby residues? And what can we learn from that information? And what we can learn is that we can start thinking about a reaction mechanism because we have ideas of how GVL or intermediates might be situated in that active site. What we came up with is a proposed mechanism that relies heavily on D229 from my mutational data and also from the structural data, and this water molecule that we think is attacking the lactone, and the calcium that's really sitting there to kind of pull everything together and get everything into that central location for catalysis. So again, we have D229 deprotonating a water-bound calcium, or sorry, a calcium-bound water molecule. The water attacks our substrate, forms a tetrahedral intermediate, and then that tetrahedral intermediate breaks the lactone ring, stealing that proton back from D229 to form product and allow for repetition of the catalytic cycle. So this was interesting to us, mainly to me because other related lactanases that have the calcium here, it has some similar active sites, don't describe this aspartate residue as being a key catalytic residue. So it seemed like either we are, onto something new and we're looking at an enzyme that does things a little differently than some other enzymes. Or what was interesting that I looked at is when we align the structures of these different lactanases from different domains of life, when we align the active sites, all of these different lactanase enzymes have that aspartate and glutamate residue arranged in the same orientation around the calcium. So some of the other residues nearby are different and have different identities, but that aspartate and glutamate are always conserved, whether it's the human enzyme or this bacterial enzyme or gram-positive enzyme. So that was really interesting to us and suggests that the counterpart of D229 in my enzyme could be playing a role in all of the mechanisms of these other enzymes, even though that hasn't been described as such in previous work. So what? I showed you this enzyme work. Why do we care? So it's important to think about this. And again, I want to bring us to what is GVL all about in the first place. We started this work thinking about GVL because it's a useful way to depolymerize biomass and get us closer to renewable sources of fuels or other chemicals. However, residual GVL is a problem and we needed to get rid of it. So now with this work that we have on this enzyme that can hydrolyze GVL, and with this work we have that shows us structural insights in how that hydrolysis might function, we have information that could be used to improve GVL hydrolysis of this enzyme or other enzymes to help us maybe treat deconstructed biomass to remove that GVL or otherwise take care of it to help us create sustainable fuels or other products. So I want to thank a number of people for helping me do this work. Especially everyone in the Donahue and Nagira labs has helped me think about experiments, plan experiments, troubleshoot things. Also in the Fox lab, Brian Fox and Craig Bingman have been a great help. Again, Craig did all the crystallography work, so super grateful for that. And if any of you need more happiness in your days, follow Troy and Deja on Instagram. There's a lot of great dog pictures that are fun. So with that, I will take any questions. Andrew.
At the end there, the overlaid catalytic site, do all of those enzymes that you showed, they have similar or comparable activity on GVL or?
Yes, yeah, that's a good question. So I think only one of them has been tested specifically on GVL. And we find that the catalysis rate is similar, but the binding is, the other enzyme binds GVL much more tightly than our enzyme does, which we think has different structural reasons, but the catalytic rate is similar.
Yeah, just wondering if there's like differences between them, why that might be their work.
Yeah, definitely an interesting thing to think about. And the catalytic rate for pretty much all of the substrates we look at is always within like one order of magnitude. So catalytic rate doesn't vary much between substrates, but the binding affinity does vary a lot between those different substrates we tested. Thanks, Andrew. Any other questions? Jeanette.
So you mentioned identifying the genes involved in the different steps in this GVL degradation pathway. once you have a set of genes that essentially cover all steps in the pathway, how are you going to use those to help deal with that?
Sure, yeah. So I talked today a lot about what we think is step one in GVL metabolism, so the hydrolysis of that lactone. But another part of my project is figuring out the rest of the steps that my favorite bacterium uses to degrade this chemical. And the idea is once we know the genes involved, we'd be able to say, put those genes on a plasmid that then you could transform into your favorite industrial microbe to allow it to break down GVL to remove that toxicity effect, but also allow it to assimilate the carbon in GVL into its own products. So the end goal is to have a portable plasmid with all of the genes required for metabolism that you can incorporate into whatever microbe you happen to be working with. Any other questions? Yeah.
Sorry if I missed that. So I'm just wondering, so you found the set of target that might be catalyzing this reaction, and you like knock out that gene in your favorite bacteria. That's the most efficient. So divide without that enzyme.
Yeah, that's really interesting. So I did delete this lactonase gene, and it appears it's not the only one. So when I delete this gene, the cells can still grow on GVL, and they don't seem to show any growth defect. And I can even test, and when I test the cell extracts from these mutant cultures, I still detect some GVL hydrolysis activity, about a quarter of what I detect in wild type. So there's probably, or there almost certainly is another enzyme that can also hydrolyze GVL. But based on this, I may have found the most active version. And it might be interesting to pursue what the other enzyme might be that's doing this. But I haven't looked into that more. I wouldn't say significantly faster. Yeah. All right. Well, thank you all. Here, Chris, we can talk after. Thank you all for coming. Thanks for listening to our talks.